in the Aiso Yufeng Li Family Department of Chemical and Nanoengineering at the University of California, San Diego. He is also the founding director of the Biomaterials and Tissue Engineering Center at UCSD. Before joining UCSD, Dr. Chen had been a professor and a Henderson Centennial Endowed Faculty Fellow in Engineering at the University of Texas at Austin from 2001 to 2010. Between 2008 and 2010, he served as the program director for nanomanufacturing at NSF. His research is in the areas including 3D printing, bioprinting, biomaterials and nanomaterials, stem cell and regenerative medicine, and he's published over 190 papers in top journals. He has received NSF Career Award, ONR Young Investigator Award, and NIH Edward Nagy New Investigator Award. He is also an elected fellow of the U.S. National Academy of Inventors and a member of the European Academy of Sciences and Arts, fellow of AAAS, AIMBE, ASME, and SBIE. He was awarded a Bright Fellow of NSF to advance 3D manufacturing techniques in 2022. To commercialize his bioinks and bioprinting technologies, Dr. Chen co-founded Allegro 3D Inc. in 2016. Allegro was later acquired by Bico and merged with Selling in 2022. The floor is yours, Professor.
Thank you, Professor Dockman. It's always great to see you all. And I know that Terasaki has been doing very well under the leadership of Professor Ali Karam-Hassani and you guys. So congratulations. For today's talk, I will touch some design innovations over the years that we have explored for digital light processing-based bioprinting and tissue engineering. First, I'd like to disclose my financial interest that we founded Allegro 3D to commercialize the bioinks and bioprinters. As Professor Dockman mentioned, Allegro 3D has been acquired by Bico and merged with CellLink. So for today's talk, I'll first give a brief introduction and our motivation about the work. And then I will touch some design innovations that we explored for DLP bioprinting. Then I will showcase some bioengineering, tissue engineering applications, including nerve tissue engineering, liver fabrication, and heart regeneration. Then I will summarize my talk. So our first motivation is really the regenerative medicine, try to repair or regenerate these damaged or diseased tissue or organs, anywhere from skin, bone, and now we are more focused on soft tissues like heart, liver, kidney, and nerves. The other important motivation is drug discovery in terms of testing these drug compounds for helping these pharmaceutical companies to develop drugs. As you know, this is a typical chart telling the years, the time, and also money spent for each drug developed approved by FDA. They often start with 2D petri dish with millions of compounds tested for human cells, but in two-dimensional, very simple one. And then if they found some good ones, then you can move on to animal testing in preclinical setting. And then if you're lucky enough, you may have a couple of them to go through clinical trials, phase 1, 2, and 3. So as you can see, this process on average, it takes 12 years and $2 billion for one drug. So this is a very important problem.
1 second, the screen disappeared.
Oh.
I just see you, Shaochen.
No, I see the screen. It might be your view, Mehmet.
Okay.
Okay.
Sorry about that.
Yeah, I wonder if other people on the internet can still see it.
It's fine. It's fine. I clicked on.
Okay. Yeah. So the challenge, lies in the starting point, because the 2D human tissue model often do not replicate the 3-dimensional dynamic in vivo phenomena. And then when they move to animal models, for the prediction of the drug toxicity or efficacy. As we know, animals are different from human. So they don't often also predict very well the toxicity. So the goal is can we actually develop these 3D human tissue models? So they carry the human cells, but they are, they also have 3D architecture to mimic the tissue organ that we are interested. And that will be really interesting, that we can hope to reduce the cost, reduce the time for testing. So I built my lab in terms of research topics with 3 areas. One is what type of biomaterials that we need to use to grow this human tissue. Then, what type of manufacturing technologies? I was trained as a manufacturing engineer to shape these material into the structure, because they are soft often, and also we are dealing with the living cells, right? So it's different from traditional manufacturing. People cutting steels, people cut steels, ceramics or plastic. They're a totally different environment, right, in terms of manufacturing setting. So we are interested in developing these bioprinting technologies to shape these materials and make them into models that can be either used for in vivo studies or in vitro applications. And at the UCD, we are very strong in both engineering and medicine. So that's a very nice setting for my lab to grow. Talking about 3D printing, bioprinting, originally you will see there are 3 major approaches. One is inkjet printing, that by using a inkjet head to deliver these droplets one by one and form these 3D structures. Or people have used this micro extrusion-based bioprinter that can use mechanical forces to squeeze out all these bio-ink with cells and gel, and to form 3D structures. And then there's another approach based on light. That's something that we explored as well, very early time, to polymerize these liquid materials. And if there's a cell in it, you can form 3D structures. But as you can see, these are all very straightforward technology, can get you the structure you need. But the issue also are very obvious. They are often slow, right? When you form 3D structure drop by drop or line by line. And in the case of light, you also going through the scanning process. So trained as a mechanical engineer, I often wonder, if we want to build a 3-hole structures, do we have to form, deposit these droplets or lines one by one, or can we learn from semiconductor industry? They often do projection manufacturing, that the structure is formed simultaneously in one layer. So that will really increase the throughput, but also with additional benefit that this kind of structure, you don't see these artificial interfaces, not by design, but the manufacturing process. So that was the motivation almost 20 years ago when I started as an assistant professor at the University of Texas, Austin. We built the first DMD digital micromirror array-based printer by just taking a computer projector. You can see that's a BenQ projector. I'm not sure if you can still buy that from the market. Then you need a software. I'm not a software engineer, but we could use the PowerPoint file. They can project these slides one by one. So that's what we put together. I think it's a good for assistant professor to have very low budget, but can still make a machine working. So, so with this idea, you can project the image one layer at a time. Once one layer is done, then you lower the stage and wait for the surface to be wetted, and then build a second layer. So you do this layer by layer processing. And it has been very powerful. We build different types of scaffolds using hydrogel materials, like these ones published back in 2006. And then also this, three-dimensional scaffolds like we use for nerve guidance, called nerve conduits, with a straight channels in the middle, or even branch structure. Because for nerve, you often see, they branch out. So can we replicate that kind of scaffold to repair nerves in that case? But if you watch carefully, you can see that there are still these lines we call artificial interfaces because the way we build it, right? Because we build one layer at a time, and the second layer was waited until the surface is wet. So that caused these issues. And it was okay for most of the scaffold, but if you use this as a medical implant, then this interface could be the failing part when they're experiencing forces. So that was an issue that bothered us for many years. And one day we figured that if we design the machine upside down, then we could do this so-called layer by layer, but continuous printing process. So let me show you this video here. So now, this is upside down design. We have a probe here going down, and these are the gel. So the light coming from the bottom, you polymerize one layer, and then so this becomes solid, you can pull it up. But in that process, this bottom surface is still immersed in the solution. So you don't need to wait for this layer to be wetted in the earlier case. So you can keep pulling, keep polymerizing, keep polymerizing until you get your device done. And the nice thing about this one is, of course, the speed is much faster. You don't need to wait for the surface, for the liquid to wet the surface for printing the second layer. But I think the most important benefit is that you can eliminate this artificial interface between different layers when you try to form three-dimensional structure. So you can see in this case, the whole structure is so smooth, you don't really observe all these artificial interfaces. That's a significant benefit in terms of structure integrity for the scaffold they are trying to put inside the body or the animal if it experiences forces, right? So it won't break. So, and you can see because of this continuous forming process, the structure, the contour is so smooth. You don't really see. This is micro scale structures. You can see it's so smooth. And I think most important application of this is when you try to build tubes and other stuff, you don't want to see this artificial interface that could leak the liquid, right? So we demonstrate this idea using this mouse aorta model. You can see that we can print this tube in a very smooth fashion, and it can also build this small outlets here just to mimic the aorta tissue in the animal. So then this is great. So we have been exploring this for many years. And then later on, when we look at this application for drug testing, pharmaceutical industry, we were often told that they work with 96 well plates or well plates. They don't do it in just one glass slice or one well. So that was a very important lesson I learned over the years. When we design the process or devices, we really need to listen to the industrial people. And then we came back with a solution. When we learn the problem, we come up with a solution. That's what an engineer can do. So we developed this so-called immersion bioprinting technology that allows you to print the structure directly in the well using this projection printing method and in a continuous fashion. So the nice thing about this, the high-speed printing, is that you can print these micro wells within a couple of seconds. So if you are looking at 96 well plate, you could finish the printing within 30 minutes. If you do this using traditional extrusion-based process, if each well takes half an hour, then for 96, it will take 3 days, right? So that's too long for them. They will never adopt that kind of technology. But if you can print them quickly for each well, then they will like it. So this, you can see the prototypes we did to build this structure in each wells. And within one well, let me show you the video here. So within one well, we can create very compact structure in this continuous flashing mode. Okay, and this is a vascular network, and it's printed within 11 seconds. Right, 11 seconds. So after this is done, you move to the second well. Depending on what image you have, you can print different structure, or you can repeat the same structure. And so this is particularly useful for printing vascular network because, they have size difference from large to small. Some of them can be as small as a couple of microns, right? So that will be very difficult to do with extrusion printing. But with the light-based projection printing, you can print these network simultaneously, so they can actually support each other if the overall structure is so weak. Otherwise, if you do extrusion printing from left to right, they already, they're deformed, right? So that makes this technology more meaningful for industrial applications. So we were able to do this through our startup company, Alaga 3D, and make into a commercial product. So now you have this machine automatic. It's no longer a commercial projector. You remodel them and then use PowerPoint slide software to do that. We have the automatic loading, automatic software to print these different wells, and you could have micron level printing resolution. You can do multiple materials, and it's built so small it fit into the bio hood, so you don't worry about contamination from the environment. Particularly when you try to build structure based on living cells like stem cells. So over the years, we also work on materials, right? So materials is important. As I mentioned in the first slide, couple of slides, our group is interested in what material can be used for tissue engineering and also be printed by our platform. So we have worked on material like a polyethylene glycol, diacrylate, gelatin that we learned a lot from Ali's lab, and then hyaluronic acid, alginate, and even decellulose material. So all these materials can be printed with this projection-based printing platform, right? Just give you some basic concept. Here you have the uncross-linked monomers. Since it's in solution, you could have different stuff to be included, of course, cells. And then you can also include other things like direct release particles, other proteins, and then when you mix them together, when you shine light, you can cross-link these areas based on the light pattern. Okay, so now through the cross-linking, you build a living construct that containing all these functional components. Okay, using gelatin methacrylate as an example. Here is the gelatin. You methacrylate them. You have these actual arms. So if you shine it with a light, you can cross-link these arms and make it into a polymer network. Okay, so now the idea of mixing these things differently that enables unlimited design innovations here, because you can use different cells, you can use different gels, you can also use different micro elements in the system. So you can combine with them with all different design, and then also the printing process itself allows you to control the material properties, specifically like extracellular matrix stiffness, which is the important part of the material property for these tissue scaffold or tissue models, right? So, and the printing itself allow you to do that. You can, have different gels with different molecular weight, or you can control the percentage of this gel in the solution to get you different stiffness, or even you can print, control this during the flight. That means you can control the light intensity and exposure time, that will give you the different cross-linking density, and as a result, you will get a different extracellular matrix stiffness. So we have done that, for instance, in JAMA, if you have different concentration of JAMA, your stiffness is different from a couple kilopascal all the way to 15 or even 20 kilopascal. So that's a big range. You can do that. That will also change the dynamics between cell interaction with that kind of environment, right? Or you can change the exposure time. In this case, you also have different control from couple kilopascal to 15 kilopascals. So morphologically, you can see the difference, even same shape, same materials, but different stiffness when you control them by different printing parameters, right? And these stiffness actually are quite correlate with disease state, right? In this soft case, that's very traditional matrigel state. Medium case, that's very much related to healthy liver on the order of couple of kilopascal, 7-8 kilopascal. Or if it's very stiffer material, then you're looking at disease models that the matrix is stiffened, right? So those can be done by just controlling the printing parameters. You can also print these different stiffness on the same platform. That allows you to really control these cells within the same materials, same culture media, but under different stiffness. So we published in this paper showing that the cell migrates differently if they are embedded in a different stiffness materials. Even though the chemistry, the material is the same, the bio-ink is the same, but the migration capability is quite different when they experience different stiffness. Okay, and more recently, we look at the issue when you try to embed to encapsulate these different materials into the ink, particularly in this case, if you have more cells being embedded in the bio-ink, the issue is if these cells are so high density, then it's very difficult to print them. So here you can see that's our design. We choose these patterns based on the idea that they're going to scatter light. So without cells, this gel is very transparent. So there's really little scattering in the ink when light is on it. But if you embed 100 million cells per mil into the bio-ink, then they scatter the light so much you could barely resolve the structure you want to print. So it's not that you cannot, sometimes you're not able to form the structures. So the main reason is that if you have high density cells in the bio-ink, they scatter the light, scatter incoming light from the printer, right? So the reason for scattering is because the difference between refractive index between the cytoplasm and also the bio-ink. Typically for JAMA, the refractive index is about 1.33, but for cell it's 1.38. So there's a mismatch, okay? So that's a challenge, right? And as a matter of fact, for printing real tissue organs, we often want high cell density because they are more closely mimicking the physiological conditions. But due to these scattering issues, as you see from most of the work in the literature now, cell density is limited to a couple of million cells per mil. And in physiological condition, we are talking about sub-billion cells per mil, right? So it's order of magnitude difference from the ideal situation. So how can we solve this problem? Well, one way of creative thinking is to learn from others, learn from different fields. So we look at scattering problems. And in other fields like CT scan or imaging or X-rays, they have the same problem, but they managed to solve the issue using so-called this contrast agent like iodixonal. When they add this iodixonal into the system, they can improve their CT scan imaging or X-ray imaging. So, and this material is now FDA approved. So we thought, why can we try this one? If we add this one to the bioink, so you can really improve the refractive index mismatching problem, and then we print. So here you can see same situation like we before. If we want to print 100 million cells per mil without this iodixonal in the bioink, you have issues, right? You don't resolve the pattern. Well, if you add certain percentage of iodixonal in the bioink, you can see even though we have still have the 100 million cell per mil, but we are able to resolve all these little details, and then print the structures. But so that's really great. But on the other hand, we also want to make sure that adding this material will not compromise the viability of these cells, right? So that's important. So we test out this different combination of the cells and the gel, in this case, in terms of cell viability. So here you see a HUVEC cell in JOMA, and these are the human strong cells in the hyaluronic acid. And this is C2C12 cells in the algae materials. And our results show that adding this iodixonal in the bioink did not affect the cell viability significantly. So it's doable. So after that, we were able to print a thick tissue that measures a couple of millimeters thick and 18 or 17 millimeter long. And we print vasculature network inside this very thick tissue, because that's what we are trying, want to do in terms of developing organs, right? Larger scale, high cell density with embedded vasculature network. Otherwise, without the vascular network, the cell will die, right? Diffusion limit. That's very obvious. So with the help of this iodixonal contrast agent in the bioink, we were able to create this kind of vasculature, and we test out with these cells, and then can see and etherealize the tubes formed in the structure. Right? So so with that, I would like to move to different tissue engineering applications using this bioprinting platform. So we'll start with nerves. Here, as I mentioned that, for nerve, you use as an implant, you put it in the body, and they often experience forces. Well, if you use this continuous printing approach, you don't see these artificial interfaces, and you can build very strong, mechanically strong conduits to guide these nerves. Okay, so this is straight one. And in this case, we have a sleeve on the side, and then 4 microchannels in the middle to guide the neurons. And we were also able to print these branch structures. And to scale up, for future translation, and we were able to build this kind of hydrogel-based materials with, 5-6 centimeter long. Okay, and that really, it's doable with this type of printing technology, because of the structure integrity is much improved compared to traditional layer by layer or line by line process. Okay. So we did experiment with a mouse. So here you can see the shiny nerves before we cut it down. This is a sciatic nerve in the mouse, and then when this nerve is gone, we put this scaffolds with linear channels in the middle, and use biological groove to connect these 2 broken ends. and then seal the wound and let the animal to heal and regrow this nerve. So you can see 11 weeks later, these nerves grow straight into the channel and make the two ends connected. Okay. And we test out this sample by taking out and look at whether they go through these different channels. So you can see this different cross section. This is the first is starting point. This 2 millimeter from the starting point of the tube, 4 millimeter and 6 millimeters. So you can see the nerves continue to grow along these channels. So that's a verification in terms of this study. And then we had an animal study here. This is the mouse with a broken left leg. Here you can see it really couldn't push due to the damage of the nerve. But same mouse, 8 weeks later, you can see it can very much recover from this nerve damage. Okay. So yeah, so with the success of this peripheral nerve regeneration, we were looking at a bigger problem, which is spinal cord injury. And we try to replicate the same idea using scaffolds like this one. And particularly here, we try to mimic the physiological, architecture with a gray metal in the middle, and then we design these linear channels on the side, so to guide these axons into the scaffolds. So we were hoping this scaffold will work. And the nice thing about 3D printing is you can take any data, a digital image, right? Like this MRI image of the damaged spinal cord, and replicate a precise copy of that and put it back, right? So you can see that's the printed structures. And we tested this scaffold, and I'll show you the result later. It didn't work well. So on the other hand, we were looking at other approaches that people use a stem cell therapy to repair these damaged spinal cord by injecting these neuron stem cells into the damaged site, and that didn't work as well. And people figure maybe because when you inject these cells into them site, they often don't have good organization among themselves, and when they aggregate, they often die if they don't have enough nutrition, right? Diffusion limit. So we thought, well, for this one, single scaffold didn't work, and therapy by injection doesn't work. Can we combine these two, right? So combination is the innovation, right? So we were able to still build the scaffolds, but now we stuff these scaffolds with neuron stem cells. So you can see these green cells stuffed into these open channels. And now when you put these scaffolds with living cells into the damaged site, and we were able to see quite difference in terms of the behavior of this implant. So here you can see all these neuron stem cell field cross these linear channels. And then over time, you can see this red axons grow rear into it because of the physical guidance and the chemical cues in the system. So that combination gave us a much better BBB score for the for the testing in rats. So here the red one is empty scaffold. You can see it really doesn't improve in terms of BBB score, and the injection with the neuron stem cell also have very small improvement in terms of the BBB score, but the combination of these two make a big difference. So so that was published a couple of years ago, and it was reported by NIH director's blog. So that's nerve. And the second important tissue or organ we're interested is liver, right? Because for most of this drug testing, you want to make sure there's no liver toxicity, whether this is a cancer drug or heart disease drug. So it's very important. Of course, the other issue is liver cancer, carcinoma. And if we can recreate this liver tissue organ, then we could potentially repair damaged liver liver organs in human in the future. So with this is in mind, we start to print livers, and particularly we want to print this liver tissue with induced pluripotent stem cell derived liver cells, hepatic cells. So, but before that, we look at what structure, what architecture we want to replicate, right? So you look at the physiological images, and figure out what pattern you need to have. They are quite complex. And they can see in the liver, you have multiple cell types, multiple blood supplies. Can we also mimic these, right? Because that's quite related to the function. And also what material you're going to use. So we developed this so-called two-step bioprinting process. Step one is to print the hepatic cells. These are iPSC-derived hepatic cells. And we use a gelatin methacrate, GELMA, and just put it in the solution, mix them, and give the pattern. This is the hexagon patterns. And couple of seconds, we can print this this liver tissue here. And now you have all these dark areas open, and we wash them out, and we put a second type of cells here, specifically HUVEC cells, and we add some adipose-derived stem cell to help the HUVEC cells in this. And also we slightly change the bioink in this. We add hyaluronic acid to the GELMA to support the HUVEC cells. So that's another couple of seconds exposure, you print this part. So after 2 steps, you have this biomimetic hepatic construct. Here measures about 3 millimeter by 3 millimeters and about 300 micron thick, okay? And with both green for hepatic cells and red for HUVEC cells with some adipose-derived stem cells. So we measure the key gene to make sure that this is a functional liver. So here in doing so, we also compare our, we call tricultural model with simple model that other people do in the old time. So we look at a 2D monolayer, 2D petri dish monolayer. We also compare to single cell model that the structure is 3D, but it contains only the hepatic cells without supporting cells. And our results show that these, major genes, albumin, HNF4 and TTR, the 3D tricultural model has much higher expression level compared to the other two. And this FFP has a gradual reduction, that's a fetal hepatic marker. So this is great. Then we also test out in terms of anabolic and the catabolic function of the liver. And again, we compare this with our with 2D models and the 3D simple models. As you can see, over the time after we printed, we can see they survive for a couple of weeks, and also the 3D tricultural model has high level of albumin secretion level compared to the other two. Same thing for the urine production. Okay, so most recently we actually have better data in terms of long-term culture, because at that time, these iPSCs probably is not mature enough, and there's also not in a optimized condition. So you can see they degrade over time. But nevertheless, the tricultural model show better function level compared to the other two. And we also explore this for testing purpose. We pick this antibiotics called rifampicin, and we compare this to the control, and also compare this with the other two 2D monolayer and the 3D simple model, single cell models. And for these 3 major CYPs, 3A4, 2C9, and 2C19, the tricultural model has much higher level of expression compared to the control, while the other two has similar results, so not much big difference. So there's a really benefit if you go multi-cell cultural model, and if you also go to three-dimensional architecture to improve the drug sensitivity. Okay. Next, I'd like to talk about the heart, because that's very important organ, and also there's so much disease associated with that for the heart. So, and if you embed these cardiomyocytes in the hydrogel, you can see a couple of days later they start to beat, right? So that's the nature of cardiomyocytes. And here we just did a simple engineering by patterning them with the lines. You can see the behavior is quite different. This is just like you often see in a petri dish. But here you can see they are much better organized compared to these randomly packed cells. There's a difference in terms of alignment. So that allows us to look at a better model that engineered, or we call instrument heart on a cantilever model, that we can design the cardiac tissue in the aligned fashion. Then we also build them along two cantilevers. One is very thin, the other is very sturdy. So when they beat, then this thin one will experience forces, and the force will translate into deformation. So for this cantilever beam, we could have an equation to describe the force and the displacement, right? So from the displacement, we can measure force. Of course, this model also allows you to see the beating frequency. So we call this instrumented heart on a cantilever model. And here you can see that after culturing for a couple of days, they start to beat. And you can see this thin cantilever was bended during beating. So that deformation will tell you the force. And if you zoom in, you can see, this is a very visible deformation, and that's related to the force generated by this tissue. So, and we were able to test out different drugs in the system. So here you have isoproterenol that was used to treat bradycardia for slow heart. And we also test others like LEVO and OM. For all three, you can see the treated cardiac tissue has higher displacement value compared to the control that has no treatment, okay? So we can use that displacement to map out the force that generated by this cardiac tissue. So that's actually quite convenient, in terms of testing purpose. You can build the heart tissue, align them, and then also build on the sensor, but basically force sensor, to give you the measurement. If you have this in a petri dish, you don't have that value, right? So that's a really beauty of this kind of heart on a chip or heart on a device, right? So then, very recently, we were approached by our collaborators in cardiology department, and they are studying developmental biology. How does the heart form, starting from little tube to have these 4 chambers? How does a heart wall form over the time? So, and we particularly want to ask, what kind of signal pathway, that help to drive this wall building, or in other words, how these cardiomyocytes grow or migrate towards the neighboring areas to form this thick heart, right? So, so that's a very good biological question, but engineering-wise, we can design a model to answer that kind of question. Here you have this we call finger type of structures. We have ventral cardiomyocytes, iPS derived here in this case. And then we put another 2 layers to allow us to control the environment in the case if we put these signal pathways like SEMA 3C protein or SEMA 3D protein, see how this design will guide these cardiomyocytes, whether they can migrate towards this direction of this signal control, right? So that's a model we built. And here you can see the results. In this case, for this row, you have CMA 3A embedded in these tiers. And then you have also CMA 3D, CMA 6A, 6Bs. And you can see actually the quite difference. For CMA 3C case, these cardiomyocytes just migrate all the way through tier 1, the second layer, the first layer, and tier 2, the second layer, that are embedded with CMA 3A. But for it, this phenomenon didn't happen when you embed other proteins like 3D, CMA 6A, and CMA 6B. So this is a very simple model that we can build using bioprinting, but answer a big biological questions. How does this cardiomyocytes migrates in that kind of environment? So that was very recent published. There's a video here also associated with this paper. So I think I'm out of time. So to summarize my talk, I think designs in innovation design can lead to very powerful manufacturing tools. In this case, bioprinting technologies that can really do high-speed printing and small resolution. Because for cell printing, the speed is very important. You don't want to have these cells outside the culture for too long, right? The faster, the better. You want to have good structural integrity, particularly if this is implant, and that implant will experience forces if it's in the body, right? So that can be done with this kind of so-called continuous printing approach. You want to have a 3D microstructures, shapes, porosities, defined material composition, so you can switch around the materials, and also controlling the material property even during the print, right? You can change the printing intensity, light intensity, to control different stiffness, so you can build a gradient along the way of these 3D structures. You can put different cell types, so you have a multicellular architecture in your tissue or organs. And here for direct in-well printing, that's a great help to the pharmaceutical industry. They can test out these tissues with different compounds in a well plate format. We demonstrate this with the 96 well plate. That's very important for them. And then the high density, that makes more feasible for future organ printing, because you do need to have high density in the tissue that you are building. And with the help of this refractive index turning agent, you can do that. So we were able to build these in vitro and in vivo models for disease studies, drug screening, or even therapeutics. So I'd like to acknowledge, the work done by generation of students. Many of them are either in academic, like Carlos and a couple other places, and also in the industries. We have student Wei Zhu become the CTO of CellLink. We have other students working also for other high-tech companies here. And currently we maintain over 10 trainees plus 10 undergrad students every year. So the undergrads are so excited to see the beating heart on their hands when they printed them. And as I mentioned, at the UCD, we have excellent medical school and the engineering school, so that makes work like this much easier. So, but also very important, I want to thank the sponsors, from early day in NSF, ONR, and nowadays more from NIH and some foundations. We've been lucky to work with multiple grants from different NIH institutes, NIBIB, Cancer Center, Cancer Institute, NEI, NINS, Muscle, and even Children's Health Institutes. And we also are supported by some individual grants from different agencies. So With that, I would like to conclude my presentation. I'll be happy to answer any question you may have. Thank you.
Thank you for the fantastic talk, Professor Chen. So there's multiple questions came in. One of them is, great talk. I'm curious whether you can recapitulate the blood vessel formation in artificial liver model.
That's always an ongoing project in the lab. You know, we published a couple of paper, including this Science Advance paper recently published, that we show with the help of this index refractive matching agent that you can build this, you can print this vasculature network inside this thick tissue, so directly, and then you can perfuse the endothelial cells into this the network to build the vasculature system. Many other people use 3D printing structure and then try to dissolve this structure and form the tube. So that's kind of sacrificial process. But here we can print directly these network in the thick tissue. We also done work several years ago in terms of so-called pre-vascularized technology that you can actually pre-vascularize the tissue and then perfuse it and form this network. So, but again, I think that's a good question. It's a very ongoing project. I think it's also ongoing project for many other labs. Try to build network, vasculature network support for a large scale tissue or organ fabrication.
Thank you. Thank you. Another question came in. What are the main challenges in human tissue printing now?
I would say, cell density is an issue. Density is that includes two, right? One is if we saw one of them already, by using matching of the refractive index, so you can print high density tissue or organ that's mimicking the physiological condition. The other is the cell production. How, because when you build this large organ or tissue, you need a lot of cells. At this moment, for doing that, it's quite expensive. So for many people, including ourselves, working on tissue printing, we focus on small one because of the financial limit. But if we were given large budget, we could build these thick, high density cell tissue or organs. And as a matter of fact, you can see a couple of recent calls from ARPA-H to call for proposals that build real heart, a human scale heart, human scale liver, or human scale kidney. So I think you can see more and more results coming out from the community nowadays. So high density is one, with vascularization for this tissue organs.
Thank you. One other thing is that what type of cells people use? Is it embryonic, iPSC, or is there cell source problem for bioprinting?
So I touched a little bit in my previous response. Cell source is an important topic. Where do you get the cells? And we use couple of iPS cell lines. You can get them commercially but they're expensive. So you can also differentiate them from iPS by yourself. That takes knowledge and time, and we are doing that too. So you can get these commercial cell lines, and I think there are also cell lines for patient-derived cells available from these commercial vendors. So it depends on what organ you are printing, right?
Thank you. Is there a possibility that when you implant the scaffold into the body, the scaffold could be contaminated with a mix of cells? How do you control surrounding cells from invading the scaffold?
Could you, what's the first part? Could you repeat?
Is there a possibility that when you implant the scaffold into the body, the scaffold could become contaminated with a mix of cells?
Oh, so that's also a good question. When we print them, we often have several steps to make sure they are not contaminated. They basically have the cells we need. So that's very important. So the printing environment, the one we build for selling has a mechanism to sterilize the printing chamber. So that's one thing. And then you can also add antibiotics in the system. Sorry, my light is off. You can add antibiotics in the scaffold. Okay. Before you print, that will help. Whether you want to fence off other cell invasion, I think that's a question. Sometimes you do want them to interact with the scaffold, right? For instance, you have this vascularization support. You want to connect with a host vasculature system to provide you with the blood supply. So that's something you need. But if you want to avoid them, I think you can build a system like a material like a polyethylene glycol, it's not cell adhesive, so they don't really, they could be a good wall to fence off your scaffolds. So it depends.
Thank you. Have you ever tried to print brain organoids from stem cells?
We were interested in that. We have initiated some study, but we don't have publication yet.
Thank you. Professor, suppose if we are going to bioprint large structures within well place, then cell settling is a problem. How can we get uniform cell distribution throughout large structures?
That's a very, very good question. And I know you have practiced this a lot already. So yes, these cells often sink to the bottom of the well, right? So when we are keenly aware of that. So when we design this Bionova X printer, we have a mechanism. So before print, we actually have this probe to go up and down for a couple of times to have better mixture of the printing. Then we start to quickly print them within a couple of seconds to minutes. So by that you have better cell distribution control. Otherwise, yes, they often tend to sink down.
Thank you. This is a question about whether, in addition to refraction by cells, it might be that the low polymer content per unit area for cross-linking, since most area is occupied by cells. Just wondering, what do you think?
Yeah, so scattering is a complex problem. Once you polymerize the structure, you could have physical scattering due to the structure itself. Before that, you don't have the structure before printing, right? So that's one. But we feel the mismatch between the cell and the ink is a major contributor for the scattering, and that's why when we put this additional into the system, into the bio ink, the problem was solved very significantly. But of course, as I mentioned, the scattering can come from many different ways. It's a matter of which one is the more significant. Yeah.
Thank you. One final question. It's not clear how the addition of a contrast agent can help with the actual printing of JALMA. Can you please elaborate?
addition of the additional.
Contrast agent. Are you
Oh,
using?
sorry, yeah. Yeah, so gamma, as I mentioned in the slide, the refractive index is smaller than the refractive index of the cells, right? So you have that. So adding this high refractive index contrast agent, additional, then overall they mix better and the scattering is minimized. So, but of course, in the paper, if you want, you're interested, you can look at the details. Different concentration of the contrast agent will have different overall refractive index, right? So you need to optimize the concentration of the contrast agent so that the print structure is the best. So there's an optimization process. I know that it really depends on what cells, what ink you use.
Thank you. Well, that's all the questions I have. Thank you for your time, for this great presentation, Professor.
Thank you.
Stay.